I'm excited to present to you this learning material, and the purpose of this learning material is to actually look at ways in which we can advance methodological techniques in experimental research. And this is just a simple guide to help those who are interested in conducting randomized control trial. And it will be a step-by-step guide that would actually walk you and give you more familiarity with this kind of research methodology or ways in which one can go about in conducting a more of a clinical trial. Thank you. So, a randomized control trial is a type of experimental research that produces the strongest or rigorous evidence for clinical practice. So the purpose mainly is just to test causality with optimal control. So a sample is taken from the population and is being put into both the treatment and the control group via randomization or by random selection. And it goes on further to do a follow-up of both the treatment and the control group. Then it goes on further to do a comparative analysis, and the results, we compare the results between both groups, and that comes out. So that's as simple as that with randomized control trial. So let's further look into what is a randomized control trial. So a randomized control trial is identified as the most highly sorted methodology in building the best research evidence, and it's regarded as the most, or maybe as the best in producing or predicting research evidence for clinical practice. So these trials are part of research methodologies that are conducted in an objective and systematic manner and can allow for controlled investigations into the predicted outcome or controlled phenomena. So it also allows for more of a coherent body of knowledge to be found and allows for testing of ideas or selected practice problems by controlling for one predictor or variable. So this type of research gives researchers a high possibility of causality, and is considered as part of the most powerful form of quantitative methods used, as it allows for an effective or rigorous form of controlling of variables. So let's take a look at the main characteristics of randomized control trial. So in all, there are three main characteristics. The first allows for a more controlled manipulation of the main treatment in the study, and this is regarded as the independent variable. The second allows for administration of treatment to some of the study subjects, and it's referred as the experimental or treatment group. And also it allows us to not administer the treatment to some of the study subjects, and this is referred to as the control group. Overall, the third allows for more of a random selection or a random assignment of the study subjects to either the experimental group or the control group. So there are several keywords in RCT, which is more, it looks at the control group, the treatment group, the randomization, the blinding. So we'll go further in depth into these keywords. So let's take a look at control group. The control group is the same as the placebo group, and they receive the standard drug, treatment, or therapy. The treatment group is the same as the experimental group, and they receive the experimental drug, as we put it, a treatment or therapy. And the treatment is equal and consistent among all subjects in the treatment or experimental group. So we give the same thing to every individual within that group. So the next one we'll take a look at is randomization. So in all, it's also part of the words, the keywords that many in clinical trials. So in all, subjects in the study are drawn from a reference group or a reference population by the use of clearly defined research criteria. And these selected subjects are randomly assigned to either the treatment group or the comparison group, and normally the comparison group is also referred to as the placebo group or the standard group or the control group. So it depends on which terminology the person wants to use. So the next one, that keyword I will take a look at, is blinding. Blinding refers to the procedure within the research where either the patient or those providing care to the patient, which is more of like the caregiver, the doctor or therapist, are unaware if the patient is in the experimental group or the control group. So either both are unaware. But in this case, we will go further to look at what is normally known as the double blinding. So double blinding is when neither the patient nor those providing care to the patient, that which is the caregiver, doctor, or therapist, are aware of the group the patient has been assigned to. So both ends are all unaware of it, and most of the times the data is being sent to a different person or a statistician to really analyze the data and provide the results back for more to prevent bias. So in terms of another, we will go on to look at the phases of clinical trials, which is part of normally, as we're looking at randomized control trials, and clinical trials falls under that. So in most clinical trials, this methodology has highly controlled settings or research units that are clinical, that in setting, and especially those organizations are normally used for the discovery of new drugs, therapies, or treatment. And the clinical trial goes through phases. So we have phase 1, phase 2, phase 3, and phase 4, and goes on. So in that sense, we will take a look at phase 1. Phase 1 mainly it's the initial testing of the new experimental drug or treatment, and it focuses on determining the best dose or phenomena. And this helps in identify side effects or any unwanted outcome. And this phase does not include any comparison groups or randomization. The next one that we'll take a look at is the phase 2. And this phase is the next phase after the initial phase of phase 1, and seeks to provide preliminary evidence of the drug's efficacy or whether the desired phenomena is working, and any possible side effects as determined in the phase 1 trial are identified. So in this case, this phase does not include any comparison groups or randomization, just like phase 1. So we go on further to look at phase 3. So phase 3, this is the phase which right from phase 2, and it seeks to provide a comparative definitive studies on the drug or phenomena under investigation. So here is when we look at comparison between both the treatment and the control group. And this new drug, the new experimental effects or the outcome of the phenomena are compared with another drug which is normally known as the placebo or the standard therapy or treatment or phenomena. And the goal is to find out whether the experimental drug, the therapy or treatment is more effective than the placebo drug or the standard treatment or the therapy. So in this case, this phase includes randomization or blinding or double blinding. At this stage, if the results indicate a significant effect of the intervention or therapy, the evidence is sufficient to warrant the application of the findings to clinical practice. Then it moves to phase 4. So the next phase that we will look at is phase 4. And this is the phase that occurs after significant differences has been found among different groups, or there are differences between groups, and if there are significant differences or treatment is deemed effective among population, the clinical trials will move to phase 4, where regulatory approval such as the emergency use authorization of the drug is granted. And the patients are given the drug and followed over time to identify uncommon side effects. and that tests marketing strategies, that's of ways in which we can get the drug to people to see how effective it's working among them. And this stage does not include a comparison group or randomization, and it's also just in that sense similar to phase 1 and phase 2. So the entire research design must meet the criteria set for an experimental study. And all interventions in the research must be clearly defined, and there must be a protocol for the establishment of the clinical applications in the study. The main goal here we'll take a look at is to provide scientific evidence that the proposed intervention causes the desired outcome in the population. Also, the hypothesis of the study must be designed to be definitive that the intervention causes the defined effect. So these are the references mainly that the knowledge came from, and thank you.